Today we're going to talk about lymphoidal tissue. We want to look at the general organization, the cells that are there. We want to look at fluid flow, both blood and lymph fluid. We'll have two parts. Part one has to do with lymph nodes and the gut-associated lymphoidal tissue. And part two has to do with spleen and thymus. Lymphoidal tissue, part one. Lymph nodes and gut-associated lymphoidal tissue. Our objectives are to describe the organization of lymphoidal tissue in general, the different organs that are involved, details of how the antigens are removed from the lymph and the blood, and trace the movement of cells and fluid through the lymph node and spleen. Part one has to do with lymph nodes and gut-associated lymphoidal tissue. Part two will be spleen and thymus. Now, the function of the immune system is to protect against foreign invaders into the body, to protect against that. And we can see here there's a host of foreign invaders ready to get into the body. We have worms, which are the biggest one, protozoa, fungi, bacteria, and the smallest ones would be the viruses. Influenza gets a lot of us. We've all had flu at some time or another. But the lymphoidal tissue is throughout the body. We can see here in different places with lymphoidal organs to help guard against foreign invaders of the body. Also, it protects and produces a germ-free environment of the body. So largely we are germ-free. When we cut ourselves, bacteria can go in, cause an infection. Or puncture ourselves is another thing, but largely we are germ-free inside there, and that's part of the function of the lymphoidal tissue, to keep us germ-free and protect against foreign invaders. Now, what are some examples of the immune response? One is reaction to get microorganisms, bacteria and viruses and parasites. Here we see in the appendix, lymphoidal tissue there is associated with the antigens that are exposed to the appendix. Reactions against tumor cells. We all have the immune system to provide surveillance for tumor cells and to kill tumor cells. Also, allergic reactions. Hay fever. You might know someone have hay fever or poison ivy. Autoimmune reaction, arthritis, type 1 diabetes. All those are immune responses. Even graft rejection. When someone gets a transplantation, they may reject that transplantation, and that's all a part of examples of the immune response. Now, there's two types of immune responses. One is the cell-mediated. These are T cells. T cells, specific active cells that recognize imbalanced cells. The T cells, largely from the thymus, or originate in the thymus and through there, they are primary defense against intracellular pathogens, cell-mediated response. So this is a cell itself doing the problem with its smoking gun, ready to attack cells with their intracellular pathogens. Also, there's antibody-mediated. Here we have glycoproteins that recognize and bind to antigens, and those proteins are antibodies. Antibodies, the B cells, primary defense against extracellular pathogens, intracellular versus extracellular pathogens, as B cells transform into plasma cells that produce the antibodies for the antibody-mediated response. Cell-mediated, antibody-mediated response. These are the cells, these are the products of the cells. Now, what are the role of the specific activities of antibodies? One is complement lysis. So it binds to complement and helps to get things to cause death of bacteria and things. Also, opsonization. It promotes phagocytosis. So the antibodies produced by the plasma cells bind to the antigens and make them more easy to be phagocytized by the other cell. Also, it neutralizes toxins. It binds to toxins and prevents them from being effective. It prevents microbial binding to the mucosal surface is another thing. Viral neutralization interferes with cell penetration. and so it binds to the virus, and then since it's attached to a protein, proteins can't go into the cell, and so as long as it's holding that protein, that virus is holding it, is being held by an antibody, it can't go inside. Also, antibodies are involved in degranulation of mast cells, another immune cell that's involved, and the antibodies on the surface of them react to antigens and called degranulation. So throughout the body, we have these lymphoidal organs. A thymus is a primary organ, and the bone marrow is another a primary organ. But then we have the spleen, Peyer's patches, lymph nodes throughout the body. So here we see this is the spleen and the lymph node, and we'll talk about those more. So the life cycle of the lymphocytes, they start out kind of in the yolk sac or bone marrow, and then from the thymus comes the T lymphocytes, and from the bursa equivalent, which is probably bone marrow in mammals, we get the B cells, and then those B cells, they repopulate the other cells of the body, or populate it, and then from cell division you produce more of those. So bone marrow and fetal organs are first source, and then we have primary lymphoidal organs. That is, they are to produce these lymphocytes in an antigen-independent environment, so they don't have antigens associated with them. And here we get the thymus, is where we get T cells and B cells from the bone marrow. So in addition to antigen-independent, we have an antigen-dependent. That is, the primary versus the secondary lymphoidal organs. And those organs, you want the antigen to interact with the cells. So lymph nodes, lymph nodules, and spleen are where the antigens that are going to be the bacteria or whatever they're going to kill, the viruses or parasites, you want them to interact with the cells that can respond. But the primary organs, you want them to develop independent of the antigens until they get things sorted out. And so here we see some characteristics of lymph nodes, spleen, and thymus, and the structural characteristics. All of these, you have reticular cells in these, and you have reticular fibers. Reticular fibers is type 3 collagen, and that branches and allows for cells to percolate through, as we'll see a little later on. So we concentrate on the lymph node, and here we see on slide 44 the lymph node, the different follicles within the lymph node, and the connective tissue. We have a capsule around the lymph node. Just inside the capsule, we have the subcapillary sinus. So this is a little sinus that goes around through there, all the way throughout around the node, and it's going to be going right in through there all the way around. So we also have afferent lymphatics, so that we have fluid coming in from lymph fluid coming in, getting out of the blood, going into the extracellular space, and then go into lymph vessels, and it percolates through in here, go to the subcapillary space, then it percolates through, and then ultimately it goes out through the efferent efferent lymphatic duct, enters the afferent and exit the efferent. And the efferent is the same location as where blood vessels go into the lymph node and come out from, and this is at one location, and that's known as a hilus or hilum, a single site of entry of blood vessels and exit, and also that takes the exit of the efferent lymphatic duct as well. So we can see those again on 44. We can see here where we have the efferent lymphatic going out through there, and here's the efferent and this drawing. So we have a capsule around it, subcapillary space. You have these follicles with a germinal center inside there. This is a cortex, and the medulla is what we have. So this would be the cortex and the medulla in this lymph node. Now in the lymph node, you have afferent lymphatic duct, and here's what we see. We can see there's endothelial cells lining, and we can see the lymph in there. And just below the capsule, so this is the capsule where the afferent lymphatic comes in. So it's coming in right in through there, and there, and there, and there. And Right in this space between there and there is a subcapillary sinus. This little thing just inside the sinus. Fluid comes in through there and then percolates down through the cortex, ends up going back into the to the lymphatic sinus, and then ultimately out this green line. So we have the afferent, and we can see there. There's also an efferent. That is the fluid going out, and that's through the hilus region. Right in through there is where that's located, or right in through here, the efferent lymphatic duct, which is how the lymphocytes that are in that are not in the blood vessels penetrate, get out of the lymph node, and they return to the bloodstream. So both afferent and efferent lymphatic ducts characterize the lymph node. So they have afferents and efferents. And we see the lymph node here. This is the medulla, the cortex, and the capsule. So some of these would probably be afferent lymphatic, subcapillary sinus right in through here. And then we have trabeculae, connective tissue, trabeculae that project out. We have germinal centers of a lymphoidal follicle or nodule, as we might see. And then we have the maxillary cords, which are where the cells are. And then you have maxillary sinuses in between. So you have cords, sinuses, and blood sinuses, or blood vessels in there as well. Now, if this is a lymph node, these are afferents, this is our efferent. We can see the reticular fibers that run through here. Reticular fibers form like a grapevine, a network, so that the lymphocytes can percolate down in through there. It provides a framework to keep the cells suspended in through there. And here we can see the subcapillary space. There's a capsule right there. And then the cellular framework, the reticular framework of cells that is supplemented with reticular fibers. And here we see one of those reticular cell frameworks. We can see the framework here with the scanning EM very well, and you can see the scaffolding that will occur with this cell. The cell itself provides a type of scaffolding. In addition to the cellular component, in this slide 45, we can see with a silver stain, we can see the reticular fibers. So type 3 collagen fibers that branch and are supported by the reticular cells and produced by the reticular cells, indeed form a meshwork through which the lymphocytes percolate through. So reticular fiber stroma functions as a structural framework of lymphoidal organs. Again, we can see a lymph nodule or follicle with a germinal center and the cuff around the outside of there with small lymphocytes in the cuff, and we can see medium type lymphocytes in the center. And this is a germinal center because there's mitotic activity that goes on in through. Lymph nodules have a germinal center to produce T and B lymphocytes against specific antigens each had encountered in the lymph node. So as a new cell goes in through there and migrates through the lymph node, it might encounter its antigen. If it does, it will proliferate and make more T or B lymphocytes. Now here we can see one of those germinal centers in through there. The follicles itself, you got the B lymphocytes and the follicles in the perifollicular region right on the outside of the follicle. So this is a follicle here and here. Outside, these would be the T cells and these would be the B cells that we can see there. Here we see in the germinal center some mitotic activity is going on through there. You can see the reticulum cells in through here, these big ones here with distinct nucleoli. So we're talking about a germinal center in one of these follicles, and this is in the cortex, just inside the subcapillary sinus, which is just inside the capsule itself. So the lymph node and lymphoidal tissue here, you can see the cords. So there's three basic things. There's a groups of cells in cords that we see there. There's blood sinuses that we see there, and then there's also lymphatic sinus. So this is a lymphatic sinus through here, and it's the same as this in through here, and this would be what a cord would be in between. And these blood sinuses would be the blood sinuses that we see going in through there. So in the cords, blood, we can see blood vessels, of course. There's a sinus, lymphatic sinus. Lymphatic sinus itself have reticular fibers, but not the blood vessels. They don't have reticular fibers inside them. Now, inside the cord, you can see macrophages, and you can see where debris has been fed to this lymph node, and the cells and macrophages themselves have taken up the debris, and is shown here in green. So if we look at another lymph node, the capsule on the outside, the cortex and medulla, we can see the cords going through there, and we can see the sinuses with a host of white blood cells located in the sinuses themselves. So in addition to afferent and efferent lymphatics, you also have blood vessels that go up through there. Mostly go just below the follicle itself, and right in through there you specialized blood vessels called a postcapillary high endothelial venules. And this is important because this is a source of the lymphocytes when they're recycling, for them to move out of the blood into the lymph node itself. It's a one-way trip. They go out of the blood into the lymph node, and then whenever they do, they have to get back through the efferent lymphatic. They don't re-enter the bloodstream there. They go through the efferent lymphatic. And so here we can see it's a parafollicular region. So if this is a follicle, the parafollicular region right in through there is where you have these high endothelial venules. And you can see them. So these are the high endothelial venules right in through there just below the follicle in the parafollicular region. And so here we can see a typical endothelial cell, really flattened cell like this. But here's another one with a high endothelial venules. Usually they look like the nuclei should be of cuboidal cells. So lymph node and lymphoidal tissue, we can see high endothelial venules. They have receptors for the T and B lymphocytes. One-way traffic, they leave the blood and go into the lymph node. They do not return to the blood. So here we can see them again, high endothelial venules, and that's what we see there. We see some migrating lymphocytes in the vicinity. So in our slide 44, we can see there's macrophages in the cord. We can see reticular cells, plasma cells producing antibodies, high endothelial venules. Here we can see high endothelial venules here in this book view and a scanning electron microscopic view of the same type of thing. So high endothelial cells and the venules in through there are where you see the lymphocytes get out of the blood into the lymph node. So they're located in the parafollicular region. That's where you find these. So high endothelial venules have receptors for T and B lymphocytes and they enable naive lymphocytes to move into the lymph node from the blood to find its antigen. It's part of the recycling. They keep recycling through until they find their specific antigens. It's entry about 90% of the lymphocytes into the lymph node. Others may enter through the afferent lymphatic as well. But regardless of how they came in, all of the lymphocytes exit. If they come out of the blood or come through an afferent lymphatic, they will exit through the efferent lymphatic. So here we can see again the sinuses in through there, and the sinuses and the blood flow. You have an artery, arteriole, then the capillary through the lymph node, postcapillary venules right in through there, where T and B lymphocytes go through a venule and then out through the vein. So they go in through here and then back out through the same hilus region. Lympho, you have afferent lymphatics, subcapillary sinus, then you go to the trabeculae sinuses there, through the cortex, into the medulla sinus, and then the efferent lymphatic vessel. So those are the way that the blood goes in and out, lymph comes in and then goes out. So in terms of the response, the peripheral organs are needed to get the antigen to find the responsive cell. And that's part of the lymphocyte recycling. It recycles around. So here's the lymph node, the postcapillary venules in through here. So blood's coming in through there. Some of them go out through there. They get out through the high endothelial venules into the lymph node and try to find its antigen. If it doesn't, it goes through the efferent lymphatic, back through the thoracic duct, back into the heart and through the bloodstream to recycle and find another one. That's part of the lymphocyte recycling, part of the surveillance system of the immune system. So you have recycling of the lymphocytes through the node, through the high endothelial venules, in through there, finally goes back to the thoracic duct, heart, and returns to find another lymph node to go through. Lymph node filtration allows potentially active lymphocytes to see and respond to the antigens. And if they see those antigens, so an infected, stimulated lymph node would swell with lymphocytes, because once they see their antigen, they're going to start proliferating. And whenever that occurs, it will cause the lymph node to swell. Maybe you've had that happen to you before. So in summary of the lymph node, the basic structure, you have lymphatic vessels, afferent, efferent. You have vasculature, blood vessels that go in there. The unique thing is the high endothelial venules, and you have reticular cell networks. You have reticular cells in through there. You have reticular fibers in through there. You got the cortex, medulla, sinuses taking out the fluid. And in the cortex, you have the follicles. You have the germinal centers, and a parafollicular T lymphocyte maturation and sinuses and cords. So in addition to the lymph node, which is very important in surveillance of the body, and that's one of the things they remove if you have a cancer in a certain spot, they will remove the regional lymph nodes because it's a likely source of cancer cells migrating through and may get caught in a lymph node. So in addition to that, we have the gut associated with, you got tonsils, you got those throughout the GI tract. Breyer's patches are some of the ones that you have in the gut associated process. So the gut associated lymphoidal process, we can see in slide 62, the terminal ileum. So this is muscle in through there, and here we see the Breyer's patches. Each one of these little Breyer's patches here look like Mickey Mouse ears or something in this particular case. But it has a high density of lymphocytes that you see there. These are endothelial, these are epithelial cells, simple columnar epithelium with a brush border. And then some of the cells right next to the lymphocytes, the high density of lymphocytes, we see some lymphocytes over here, but these are high density of them, are the end cells or the macrophage cells. So these are not tall cells like these. And these have a role of presenting antigens. And here we can see them. Those cells again right in through there. They're not tall like these, the goblet cell and tussle absorptive cells. These are a little bit different. And these cells, as you see in a scanning electron microscopy view, have a role in presenting antigens to the surface. The function of the M cells is to present organism particles from the gut lumen to the immune cells across the epithelial barrier. So it takes these and presents these to these guys so they can react to their antigen and then once that occurs, the cells migrate through the lymph node, through the thoracic duct, back into the bloodstream, and so the B cells would then transform as plasma cells and start producing antibodies against whatever antigens they are exposed to. Breyer's patches in the human appendix is one of those examples of lymphoidal tissue, and it could have a germinal center. In through there, you see a germinal center right here. There's more lymphatic tissue there. In the stomach, these are gastric pits that we see through there, but there's lymphoidal tissue going through there. And this is in the esophagus. So we're heading back from the gut toward the oral cavity. So this is in the esophagus. You see a germinal center and a nodule in through there. And then here comes the tonsils. Tonsils at the top. Here we see tonsils in slide 46. Tonsils in through here. These are the tonsils crypts, where food could get trapped into there. And you know it's tonsil because you got a stratified squamous epithelium. As non-keratinized stratified squamous epithelium characteristic of oral cavity, but also you can find that type of epithelium in the esophagus too. In the tongue, we have stratified squamous epithelium there, non-keratinizing as well, and you can have lymph nodules scattered throughout there as well. Here's another view of the tonsils with these crypts in between there. High density lymphoidal tissue, stratified squamous epithelium, non-keratinizing on the surface. If a patient had a tumor in the posterior third of the tongue region, the lingual lymph tonsils could be removed, and at least by check for cancerous growth as well. So this is the end of the first part. It has to do with lymph nodes and gut-associated tissues. Many illustrations in this presentation were modified from books and sources that are indicated here. We want to thank the original sources for the information that was modified for this presentations. Thank you.